Namaste Makle. Today we are going to be doing some important questions on human reproduction from NEET Prep, and we are going to be seeing about close to 20 odd questions, but you will learn much more apart from those 20 odd questions during this revision that we are going to be doing. These questions are from NEET Prep. We will quickly start with the first questions, and these are all on human reproduction. Very interesting questions, and we will be dwelling more into certain subjects as you go through. So don't miss this opportunity to learn more from RN NEET. Okay, the first question is the cleavage divisions in humans is. So they want to tell about what is the type of cleavage division that you're actually having in humans. So the first question is the cleavage divisions in humans is 1. Holoblastic, equal, intermediate. So they're talking about the type of cleavage division that you get in humans, and that means in the zygote. Once the zygote is formed, once the zygote has to start dividing, how is the division that is taking place? What are the choices given? Holoblastic, all of them it's holoblastic, so you know obviously it is holoblastic. What is the other type of division that occurs if it is not holoblastic? It is meroplastic. What is the difference between these two? We'll come to it. Then equal and indeterminate. Holoblastic, unequal, indeterminate. Holoblastic, equal, determinate. Holoblastic, unequal, determinate. So you need to pick out the right choice. So first, you know it is always holoblastic. In humans, what do you mean by holoblastic? Holoblastic basically means that when you actually have the whole zygote, if there is a division, now all of them have a pole called the animal pole and the vegetal pole. Okay, so there's the animal pole and a vegetal pole. But these are the two poles, like a north pole and south pole, and these poles are called the animal pole. What is it called? The animal pole and the vegetal pole. So between these two, when division occurs, and if the division occurs from one end to the other end and goes through completely, then that is called as the holoblastic, dividing it into two either equal parts or sometimes unequal parts. It could travel from across the whole thing, from animal pole to vegetal pole, but sometimes going like that. So now you see here there is two unequal parts. So you can have a holoblastic division with either equal or unequal. In humans, it is holoblastic and equal. So the choices are either this or this. So it is not unequal, it is equal. Okay, sometimes you do get unequal divisions which are possible, especially in amphibians. Where do you get unequal? So remember that, you can write it down. Unequal division occurs in amphibians. Okay. Now the next thing, okay, if it is not holoblastic, what are the other division that occurs? The other division that occurs is called as meroplastic. What do you mean by meroplastic? In meroplastic, the yolk sac doesn't get divided. Now, you know this particular thing, there is an embryonal disc that is there present here. In spite of that, when division occurs, it totally divides the embryonal disc. It divides this yolk sac also. Whereas in holoblastic, both are divided. Whereas in meroplastic, if you have the egg, the total zygote here, and you have the embryonal disc, and if you have the yolk sac, there is no division that occurs at yolk sac. The division occurs only up to the embryonal disc. This can be either very superficial or going up to the embryonal disc. So the meroplastic division, so this is called as the meroplastic division. This meroplastic division is either two types. One, it is either superficial or it is discoidal. Okay, the superficial type of division takes place in insects. Insects have superficial in insects, whereas discoidal is present in reptiles. Okay, so see, from this one question, we have derived so many other questions which are possible, including to say that, like, unequal division occurs in amphibians. Division is holoblastic, meaning it is coming from animal to vegetal pole. Meroblastic division means the yolk sac is not divided. Superficial division takes place in insects, whereas discoidal takes place in the reptiles. Okay, so these are all different choices that you are getting from one single question. Then the next part which is important for us is, the other thing we need to know is whether it is indeterminate or determinate. So this is a dividing point here between these two choices, whether it is indeterminate or determinate. What do you mean by determinate or indeterminate? Now, when you're basically talking about this division, as the division takes place, is the future cell, okay, so there are different cells on either side, and when the division takes place, is it that the division is taking place so that if, for example, you need this particular cell to become one hand and you need this cell to become the leg, are they dividing in such a position, in such a manner, so that this particular cell automatically then becomes the leg and this particular cell becomes the hand? One becomes a hand, the other becomes a leg. One becomes the forearm, the other becomes the hind leg. Is the determinant, is the cell already determined to become something else? Or is it indeterminate? Which means this cell can also become the leg and that cell can also become the hand. Is anything possible? So is it that the type of division that takes place, is it such that its future location, its future organ is already decided, which makes it a determined type of a division? So this is what it means when you say determinate or indeterminate. So if the fate of the blastomere, cell is predetermined, you call it determinate. If the fate of the blastosmere is not predetermined, you call it undeterminate or non-determinate. So in humans, you know that it is holoblastic, equal, and determinate. So the answer for this question is, C, which basically tells it is holoblastic, equal, and determinate in humans. So the answer is C, that is 3. Perfect. We'll now move on to the next question. The testis is situated outside the abdominal cavity within a pouch called the scrotum. This is necessary as the scrotum can contain a lengthy duct for the transfer of sperms. The scrotum helps in maintaining the low temperature of the testes necessary for spermatogenesis. The scrotum reduces the pressure around the testes necessary for spermatogenesis. Scrotum can store huge amount of sperms. You know that this is wrong. The scrotum does not store any amount of sperms. Okay, true. And all the sperms are basically stored within the epididymis and the vas deferens is not the scrotum. It contains a lengthy duct for transfer of sperms. The lengthy duct is present in the vas deferens but not for the transfer of sperms, and not necessary that is the reason. Okay, so that is also wrong. You know that scrotum helps in maintaining the low temperature, okay, that is necessary for the spermatogenesis. So this is the right answer. The scrotum reduces the pressure around the testes necessary for spermatogenesis. No. So it is basically not for reducing pressure or anything. It is for maintaining the temperature. The temperature for the amount of spermatogenesis needs to be how much? 2 degrees Celsius or 2 to 3 degrees Celsius below the body temperature. So below the body temperature of normal 37 degrees Celsius, you need to get this little 2 degrees Celsius, little lesser. and that is why the testis is maintained inside the scrotal cavity and the scrotal is present outside the abdomen. Okay, now a couple of important points and important questions that you might want to actually know from this particular important concept. Okay, so let us basically look at some important concept that you might want to know. First, okay, is the testis, okay, present and in the scrotum right during the embryonal development? Is the testis already developed in the scrotum? Is the development of the testis takes place in the scrotum? No, the development of testis takes place in the abdomen. Okay, and so therefore the testis therefore descends down to the scrotum. When does that happen? It typically happens around the 7th month. So that is when the testis tends to descend down into the scrotum. For the testis to descend into the scrotum, okay, how does it take place? Where does it descend from? There is a small canal basically. Okay, now if you know that basically this is the groin, okay, and that is the thighs. Now there is a small cavity here, okay, which is coming from the abdomen to the scrotum area, and this particular area is called the inguinal canal. What is it known as? Inguinal canal. And so therefore the scrotum descends from the abdomen into the through the inguinal canal into the scrotal sac. So this is a very important point that you need to note, that the inguinal canal is a place through which the testis will descend, and it descends in the 7th month. Okay, so important point that you got. Also, there is another important question. You know that the scrotal sac is maintained outside the human body in order to maintain the lower temperature, and so therefore it is susceptible to changes of the outside external temperature. What are the external temperatures? Very cold. Now you want it to be 2 degrees Celsius below the body temperature of 37. Now if the temperature outside is very low, you know that the normal homeostasis of the body helps you to maintain the body temperature, whereas the scrotum is now present outside. Won't that make the scrotum more susceptible to the outside temperature? Which means something inside the scrotum now needs to ensure that the testis is not susceptible to a cold temperature. In order to make it warmer, what is the mechanism it has? The scrotum has got 2 important muscles. What are those 2 muscles? Cremaster and dartus muscle. What are the names of the muscles? Cremaster and dartus muscle. These 2 muscles contract to pull the testis a little bit closer to the abdomen. So they tend to pull the testis closer to the abdomen. So in order to do that, the cremaster muscle and the dartus muscles of the scrotal sac contract and pull the testis closer to the abdomen in order to maintain its temperature closer to the body temperature in cold climates. So that's why basically the cold scrotum becomes very tough due to the cremaster and the dartus muscle contraction in cold weather. Okay, so it's an important point that you need to remember. We'll go to the next question. Each seminiferous tubule is lined on the inside by the spermatogonia, the primary spermatocyte, the Sertoli cells, and both 1 and 3. You know that the spermatogonia structure, if I take into consideration, you have got multiple lobules, and there is a interstitial space that is actually present in between the lobules, right? So there is an interstitial space between the lobules, and each of those lobules are lined by spermatogonia as well as Sertoli cells. So both spermatogonia and Sertoli cells, okay, are present which line this particular cells. So their one single lining, outside lining, is spermatogonia and Sertoli cells. Both of them are pyramidal in shape. They are typically pyramidal in shape. Okay, so 1 and 3 is the right answer. Correct? So no confusions here. But are primary spermatocytes present inside the seminiferous tubule? Yes, they are present. These particular spermatogonia will then become the primary spermatocyte, the secondary spermatocytes, and then the spermatids, which are present inside, and these spermatids are attached to the head of the spermatid is attached to the Sertoli cells, and then it tends to come out into the fluid which is bathing this particular whole follicle inside, and then that particular spermatid which is released then basically becomes the typical spermatosperm, okay, which actually enters into the vasa efferentia and then outside to the vas deferens. Okay, now there is an interstitial space which is present inside, and this interstitial space also has got certain cells, and they are known as the interstitial cells of Leydig. So I think our next question basically is about it. The next question, fourth question, we talked about the interstitial cells of Leydig. Exactly, that's what is asked here. The Leydig cells are present in the seminiferous tubules and they secrete androgens. They are present in the seminiferous tubules and help in maturation of the sperms. They are present in the interstitial space and secrete androgens, are present in the interstitial space and helps in maturation of the sperms. They are present in the interstitial space, you know that, right? So that's an important point. You know that they are present in interstitial space and they secrete the androgens. So these two are important points. So therefore the choice is C. The seminal plasma is rich in. Now you have asked about the seminal plasma, which is what does it contain? It contains sucrose, calcium, certain enzymes, glucose, sodium, certain enzymes, fructose, calcium, certain enzymes, fructose, sodium, certain enzymes. You know that the answer is specifically seminal plasma. One of the main things that is present in it is fructose, also calcium and certain enzyme. What type of enzymes? Proteolytic enzymes. Okay, they do contain proteolytic enzymes. And not important is not sodium but calcium, and also presence of fructose. Presence of fructose in semen is a very important thing because no other secretion in our body contains fructose. So therefore the presence of fructose is a very important test, forensic test of sexual assault. Okay, so that's an important point that you need to know. What are the proteolytic enzymes that are present, okay, in the sperms? What are the proteolytic enzymes? Okay, these includes mainly fibrinolysin, okay, and also other proteolytic enzymes like fibrinogenase, fibrinolysin, acid phosphatase. Now this is secreted by the prostate into the seminal fluid. So the acid phosphatase, fibrinolysin, amylase, also and pepsinogen. So these are the important enzymes, especially fibrinolysin, acid phosphatase and amylase are important enzymes which are present in the seminal plasma. Yes, children, we'll continue with the next question. The spermatids are transformed into spermatozoa by a process of. So we want to know what is it that the spermatids are transformed into a spermatozoa by a process called. What is that process by which the spermatids gets converted to spermatozoa? If you probably look at this question, there are two. That's a little tricky question. See this question. Basically, what is the process of transformation of spermatozoa by spermatids to spermatozoa. Is it spermiation? Is it spermatocytogenesis, spermiogenesis, spermatolysis? It is not lysis. There is nothing called spermatocytogenesis. It is either spermiation or spermiogenesis. So what is it that is from spermatids? Now you know that basically the primary spermatocyte becomes secondary spermatocyte and then becomes the spermatid. The spermatids then mature into spermatozoa. And what is that process known as? It is known as spermiogenesis. It is not spermiation. What is spermiation? The process by which this spermatozoa, once matured, once formed, they get buried into the head. The head buries into the Sertoli cells. What are they cells? Sertoli cells. Okay, the matured spermatozoa itself get buried into the Sertoli cells and remain there. And then inside the whole lobule, if you see, there are different spermatogonia and Sertoli cells, and then you have the spermatozoa which are buried in the Sertoli cells. The process of release of these sperms, into the lobule and then into the vasa efferentia and into the semen is called as spermiation. So the process of release of the sperms from the Sertoli cells is called as spermiation. The process of maturation or transformation from spermatids to spermatozoa is spermiogenesis. So very important to differentiate these two important aspects. This is very nicely given in your NCERT. We'll read just that. The spermatids are transformed into spermatozoa by the process called spermiogenesis. After spermiogenesis, the sperm heads become embedded in the Sertoli cells and are finally released from the seminiferous tubules by the process of spermiation. So the process of release from seminiferous tubule is known as spermiation. So differentiate these two important factors. Don't confuse the spermatids becoming spermatozoa as spermiation, that is spermiogenesis, and the other one is release of the sperms into the from the seminiferous tubules into the vasa efferentia is known as spermiation. Okay, right. Next question, what causes the onset of puberty in males? Is it increased secretion of testosterone by the testes, decreased secretion? It cannot be decreased, you know that. Increased secretion of GnRH by the hypothalamus or decreased secretion of GnRH by the hypothalamus. You know it is not decrease, it has to be an increase. Always it is an increase. So what is it that is increased? Yes, during puberty there is an increased secretion of testosterone, but what is it that causes the onset? The onset of puberty is controlled by the hypothalamus. It is not controlled by the testes, because the hypothalamus has to first cause an increase in secretion of GnRH. This is then subsequently going to act on the pituitary. The GnRH, okay, so the GnRH is released by the hypothalamus, and from there you get the gonadotropic hormone, which is going to come from the pituitary. And what are the gonadotropic hormone? The interstitial cell stimulating hormone, correct, and the follicle stimulating hormone. These will then act on the testes and cause the release of testosterone by the Leydig cells. So therefore the onset is triggered by GnRH. Remember this important point, which is also the reason for onset of secondary sexual characteristics. So the answer has to be a 3. Okay, right. Next question, for normal fertility, what percentage of sperm in ejaculation must exhibit vigorous motility? We saw this in the shorts. You've seen the orange NEET shorts. You would have got this question. We did this in the rapid fire questions also. The normal fertility, vigorous motility is, you know, the answer is what? 40%, right? So 40% is for vigorous motility. And what is it for normal morphology? 60%. So morphology is 60 and motility is 40. So combined with normal morphology and motility, if they ask, it is actually 40 into 60, which is actually 24%. So normal motility and morphology together, if you ask you together, then you remember this as 24%. So it's a very important point, and yes, this is commonly asked. Don't forget this. The division of primary oocyte results in. Okay, so let us see what is the division of the primary oocyte results in. The unequal cells of smaller haploid secondary oocyte and a larger diploid polar body, unequal cells of larger haploid secondary oocyte and a smaller haploid polar body, unequal cells of smaller diploid secondary oocyte and a larger haploid polar body, equal cells of haploid oocyte and a haploid polar body. So it's a simple question. Put it in the chat box. I would put it in the comments box. I would like to see how many of you get this answer right. It's very simple, straightforward. You know that the primary oocyte is going to give you a secondary oocyte and a smaller polar body. Correct? So it is unequal. Which is larger? The haploid secondary oocyte, okay, and a smaller haploid polar body, right? Or is it a diploid secondary oocyte and a larger haploid polar body? No, it is a haploid oocyte, secondary oocyte. So primary oocyte is diploid, secondary oocyte is haploid. So remember this important thing, that secondary oocyte is always haploid, right? So you know, you've seen the chart which I've told you, which is very important for noticing what is the changes that is there, right? So don't forget this basic chart. We have shown that in your this one also. Okay, so this chart is extremely important. इसको भूलना नहीं, हाँ, इधर मत तो पड़ी, please. Perfect. So the answer to this question will be unequal cells of larger haploid and a smaller haploid polar body. So that is answer number 2. Okay, right. The correct sequence of various menstrual phases. So the phases of the menstrual phases. This is very simple. Put it in the comment box. See the answer question and put it in the comment box. What is the right answer? I'll just wait a little bit for your responses. Very important. See, sometimes they can easily you know, alter the order, and it need not start from follicular. It could start from either you know, ovulatory, or it could start from secretory. So it need not be in the typical order that we have learned. They could just start from secretory and then go to the next phase. After secretory comes menstrual, and then comes, you know, the follicular, and then the ovulatory. So basically the order could be shuffled. So here basically it is, what is the answer? Answer 2. Why? Because it should start from menstrual, then after that comes the follicular, then the ovulatory, secretory. So ideally you can remember it as follicular, ovulatory, secretory, and then menstrual. So it should be starting from follicular, ovulatory, secretory, and then menstrual. So menstrual could come first, then in that case it is menstrual, follicular, ovulatory, secretory. Okay, right. This question is asking what happens during pregnancy. Question number 11. These are all questions from NEET Prep. We're just going through randomly few good questions, and then later on we will assemble them all together. During pregnancy, all events of the menstrual cycle stop and there is no menstruation. Is there any menstruation during pregnancy? No. Does all events of the menstruation cycle stop? That is what you need to know. Events of the menstrual cycle continue, but there is no menstruation. Is that right? Basically, no. All events of the menstrual cycle will stop. That means there is no growth, no follicular phase, no ovulation, no secretory phase, because if the secretory phase occurs, then the endometrium will break down. Then endometrium cannot hold the fertilized egg. The zygote cannot hold it. So therefore all phases, even the secretory phase, ovulatory phase, and the follicular phases, all of them stop basically. Okay, and so therefore complete different cycles of the menstruation, and there is no menstruation either. Okay, so the answer is very simple. Answer is 1. Right? Perfect. Right. He's asking, in the absence of fertilization, what happens to the corpus luteum? Okay, in the absence of fertilization, what happens to the corpus luteum? Okay, it stops secreting progesterone but increases the secretion of estrogen, stops secreting estrogen but increases the secretion of progesterone, increases the secretion of both, or degenerates. You know, in the absence of fertilization, the corpus luteum, which is generally initially, why is it called luteum? Because it is yellow in color. Because the corpus luteum basically has a pigment called lutein, and that lutein is yellow in color. That is the reason why it's called corpus luteum and secretes progesterone. You know that in the second phase. Now, once fertilization occurs, it continues to secrete progesterone. In the absence of fertilization, okay, that means fertilization is not taken place, then in that case it degenerates and becomes white. It becomes corpus albicans. That's why it's called albicans, because albicans means white. What happens? It degenerates. So the answer is just 4, that it degenerates. What ensures that only one sperm can fertilize the ovum? Okay, so monospermy. Okay, what is it that actually controls the monospermy? Now, you remember that basically around the zygote, around the ovum, basically which is to be fertilized, okay, you have an outer layer which is called as zona pellucida, and then inside that you have one more layer called the vitelliform membrane. And then outside this there are cells called the corona radiata. What is it that actually helps you to prevent polyspermy? Monospermy is facilitated or polyspermy is prevented, okay, by which of it? Is it the corona radiata? Is it the ground substance around the ovum? Is it the zona pellucida, or is it the acrosome? No, it has only the zona pellucida. The zona pellucida has got specifically proteins fertilizing and anti-fertilizing that ensure that, like, you know, there is only one sperm that can enter in. The moment one sperm enters in, there is a fertilizing and anti-fertilizing antibody formed, which creates an agglutination. So therefore, once that happens, there is no further possibility of another sperm to attach, because the agglutination, this fertilizing anti-fertilizing reaction cannot take place. The corona radiata actually helps in degenerating other sperms subsequently. But so therefore, what is important is what ensures that only one sperm can fertilize? It is the zona pellucida. So the answer is 3. This question tells us about what happens during pregnancy, the levels of thyroxine. Okay, so the levels of thyroxine, what happens? Okay, so this is interesting. Does it increase several folds? Does it increase several folds in the fetal blood? Does it decrease several folds in the maternal blood? Does it decrease several folds in the fetal blood? Okay, so what is the choice here? There is an increase. What is happening during pregnancy? There is an increase in the metabolic rate of the mother. So it has to metabolize both for the fetus as well as for the child, for the fetus as well as for themselves. So therefore, the metabolic rate has to increase. So when the metabolic rate increases, the thyroid hormones has to become more. So it becomes more not in the fetus but in the maternal blood. So it does increase several fold in the fetal, in the maternal blood, right? Okay. Done. These are all very, very important questions that you should know, because all these concepts, you know, these concepts about increase in thyroid hormone in the maternal blood, these things are all important concepts. Only when you revise these concepts will you really be able to solve a question, because they're not going to give you straightforward single questions. You're going to have multiple statement questions, and towards the later part, when you are doing your revision after January, we will be doing a simulation of all such questions, and we're going to be having a lot of mock tests during that period, right? So the first movements of the fetus and the appearance of hair on the head are usually observed during. So very important. See, the 2 parts he has given here. One is the appearance of hair on the head as well as the first movement of the fetus. What is the first movement of the fetus called? What do you call it? You call it quickening. The first movements of the fetus are known as quickening. What is it known as? Quickening. Okay. So remember that the first movement is known as quickening, and the appearance of hair on the head occurs in the 5th month. Which month? It is the 5th month. So the answer here is 5th month. Okay, so these are very important milestones that we are going to actually discuss different aspects. For example, all organs appear by which month? All the organs, all the major organs are formed by which month? All organs gets formed by the, put it in the comment box. Let me see. All organs are completely developed by which month? By the 3rd month itself. Okay, very important point. Limb digits, they ask this, limb buds or limb digits are generally formed by how many weeks? How many weeks? We said all organs by the 3rd month. So limbs can't be beyond the 3rd month. It has to be less than the 3rd month. So it should be in few weeks. How many weeks do you get limb buds? Remember, it is just like how you had appearance of hair was 5th month, whereas that is 5 weeks. So limb buds is by 5 weeks. Okay, which is another very commonly asked question. Appearance of heart. Okay, all organs you said 3rd month, but the appearance of the heart is by how early? How early do you get the presence of the cardiovascular system and the presence of heart? Heart is present by the 4th week itself. Okay, so these are all important milestones that you should remember. We'll prepare a chart. Observe the RNEET shots, because just go through the multiple shots that you see in RNEET, because you're going to catch those small important questions, and in fact sometimes questions which are difficult are posted on the shots. There was one very interesting shots on what are cells of Ruber. How many of you know that? What are cells of Ruber? If you don't know, please go and see the shorts portion. You will basically be able to easily understand in just a minute, in less than a minute, what are cells of Ruber. Okay, we'll continue. Milk produced during the initial days of lactation, called colostrum, essential for the newborn, as colostrum contains. There's a very common question asked. Colostrum is rich in which antibody? It is basically anti-infective antibody. So what is the colostrum made up of? Does it contain large amount of glucose? No, it is low amount of glucose. Remember, the glucose content in it is low. What is high? It is rich in protein. It is a highly rich protein substance. And what does it contain? Low fat. It is low in fat, very low fat. Okay, it's low fat, okay, low glucose, rich protein. And what does it contain? Hormones? No hormones. What does it contain? Increased antibody. Which antibody? Put it in the comment box. IG. Which IG? Yes, IgA. So IgA is very much present in the colostrum. So this is a very important aspect which gives immunity. What type of immunity the child gets, that also they ask. Okay, they put it in statements. Consists of IgA, which giving you active immunity. Increase of IgA giving passive immunity. Now remember here, IgA is basically produced as a part of active immunity, but the colostrum already contains it. So the child actually is directly getting IgA, is not producing the IgA. So therefore the fetus or the child, the child actually at the time of breastfeeding basically gets passive immunity due to IgA. So this is going to give you passive immunity to the child, right? Though it is secreted in the milk of the breast milk. So therefore the child directly consumes the IgA. So it is not active immunity for the child. It is a passive immunity for the child. Okay, right. All the following structures are derived from the mesoderm except the. So here we are talking about the 3 layers, of the gastrula. That is, gastrulation is basically, what is gastrulation? The process of conversion of gastrula to gastrula, wherein the different layers and their future determined structures are identified, right? So ectoderm, mesoderm and endoderm. So endoderm forms few things. You know that the gut and all that is formed by the endoderm. The mesoderm basically going to form all your connective tissue, the muscles, the blood, everything is formed by the mesoderm. And the ectoderm is basically all your superficial epithelium. Right. Okay, so that is formed by the ectoderm. We're going to put out lovely chart. Okay, this is a very lovely chart in Arihant publication, and so I'm going to put that up in the shorts, wherein you're going to see a complete division of what is formed by the ectoderm, what is formed by the mesoderm, and what is formed by the endoderm. So it's a very important chart. Please look into that. It will be very helpful. So the advantage of basically following NEET is you're not going to be seeing only questions from one aspect. Today we are doing from NEET Prep. Tomorrow we are going to be doing from some other book. Tomorrow we might be doing from say a complicated questions from Alan, Akash, or many of them. So we're going to integrate all of it together and make sure that you understand and know all the topics. If the question is already done in this and the similar question is coming in somewhere else, we're not going to be repeating that. So we're going to be only doing those questions which are important at the same time that you understand the whole topic. And so this question is basically about which structure is derived from the mesoderm except. Now mesoderm basically is what from the skeletal system. So therefore the cardiovascular system, correct, is derived from the mesoderm. So heart is there. Okay. Now, is it the dermis of the skin or the epidermis of the skin? The epidermis means superficial. So it is from the ectoderm. Dermis basically is part of the muscle tissue. Similarly, connective tissue. So dermis means it's underneath the epidermis of the skin. So it is the layer underneath the skin is called as dermis. So that is a kind of a connective tissue. If it is connective tissue, it is, remember, it is again mesoderm. Connective tissue is again mesoderm. Right? So the dermis is also mesoderm. Heart, is connective tissue, mesoderm. Is it notochord or the pancreas? Remember that the ectoderm basically forms two things. In the word ecto, E-C-T. Remember this, E-C-T, ectoderm, stands for E for all epithelium. Okay? C stands for crest. C represents crest. What is crest? The neural crest. Okay, the neural crest is coming from the ectoderm. T for tube, the neural tube. Okay, the spinal cord tube. Okay, and the brain comes from the ectoderm, neural tube. Okay, not the notochord. The cord, notochord, notochord does not come from the ectoderm, it comes from the mesoderm. So the only neural structure that comes from the mesoderm is notochord. Okay, neural crest and neural tube. Okay, these two things come from the ectoderm. It's there in the word ecto, E-C-T. Remember this, beautiful, E-C-T. So that is the advantage. You will tend to learn things like, how to basically make these shortcuts. That is what NEET will give you. Ectoderm, E for epithelium, C for crest, and T for tube, neural tube. So these three. So therefore notochord comes from the mesoderm. So this is also right. What does not derive from the mesoderm? It is the pancreas. Remember, pancreas is a part of the GIT. The liver, the gut, okay, the pancreas, the whole gut, stomach, duodenum, all of them basically, and the liver and the pancreas, all these are part derivatives of the endoderm. We will put that chart on the RN shots. Do remember to see that. The sperm has several components, out of which the function of the acrosome is 2. Okay, he's telling you several components are there as a part of the sperm in that acrosome. So you want to know what is the acrosomal reaction. Okay, so what is it basically? It has got enzymes. What is the purpose of the acrosome? Enzymes, acrolysin, right? Acrolysin, fibrinolysin are present in the acrosome. And what is that? And hyaluronidase. What are the enzymes? Acrolysin and hyaluronidase. And what is the function of it? They break down the membrane of the ovum. They help to break down the membrane of the ovum. It is not that it helps to basically propel the sperm through the fluid, is basically by the tail. Produce base to neutralize the acidic environment of the female reproductive system is by the prostatic secretions. The semen itself consists of alkaline medium, and that helps to prevent the acidic environment of the female reproductive system. The Cowper's glands, okay, you saw that, you remember that Cowper's glands and the Bartholin's glands, basically they are the accessory glands. The Cowper's gland secretes also an alkaline secretion, okay, and also mucin for lubrication. And what is their thing? They also give you alkaline. But what is the purpose of that alkaline secretion here? To neutralize the acidity of the urine, okay. They neutralize the acidity of the urine, basically because it's a common tract. The urogenital tract is a common tract, and so therefore you need to neutralize the acidity of the urine so that the semen, the spermatozoa, do not get destroyed by the acidity of the urine. And for that, basically the Cowper's glands are needed. Okay, whereas neutralizing the alkaline acidity of the female reproductive system inside the vagina is by the prostatic secretion, which is much more alkaline. Okay, and what helps to propel the sperm? It is the mitochondria as well as the sperm tail, which basically has increased motility in the presence of calcium. So presence of calcium helps in capacitation. So that is what helps to propel. Metabolize the sugars provided by the semen for energy. Metabolization is by the mitochondria. So release of the enzyme that breaks down the membrane of the ovum with enzymes. What are the enzymes? Acrolysin and hyaluronidase. Remember these two enzymes. Lovely. At what point does the secondary oocyte complete meiosis? When does the secondary oocyte complete meiosis? We know that the secondary oocyte basically undergoes meiosis. The meiosis gets arrested at the metaphase, right? And so therefore, during fertilization, there is formation of or promotion of the anaphase factor. So what do you get? Promotion of the anaphase promoting complex, APC, is increased. Okay, and metaphase arresting factor, okay, MAF, metaphase arresting factor, and the anaphase promoting complex is increased. So the metaphase gets arrested and then the anaphase gets promoted. So there is an arrest in metaphase and then the anaphase continues. So therefore the point, so at what point does the secondary oocyte complete its meiosis? It completes its meiosis the moment there is penetration by the sperm cell. So only on fertilization you know that the secondary oocyte completes its meiosis. So it's a very important point that secondary meiosis is completed at fertilization. So the answer here is 3, right? Good. I hope you people are answering the questions even before I'm telling it. Put it in the comment box, see the question and immediately try to put it in the comment box. Okay, right. How do ovulated oocytes get into the uterus? Okay, so how does it get it? Is it because the ovaries are within the uterus and the ovulated egg is already within the uterus? No, it is not within the uterus. You know that basically the egg is released where? Outside the fallopian tubule, right? It is collected by the fimbriae. You know that, right? So cilia within the oviduct create currents that sweep the egg towards the uterus. Possible. Okay, so you know this is wrong. The oocyte actively swims through the oviduct using its flagellum. Does the oocyte ever have a flagellum? Only the sperm has, not this. So therefore this is wrong. Fimbriae sweep over the oviduct. Yes, fimbriae are present. Does it sweep over the oviduct? No. Fimbriae sweep over the ovum. Sweep over the ovum. So if that was given, that fimbriae sweep over the ovum, this would have been also right. So this is also right, this is also right, but in this case it is fimbriae sweep over the oviduct, so that is wrong. It is fimbriae sweep over the ovum and tend to capture it into the fallopian tubule. And then what is it that basically helps? The fimbriae tend to sweep over it, the ovum, but it is the negative pressure within the fallopian tubule. Remember that it is the negative pressure and currents which are created because of the movement of cilia. Okay, so you know the fallopian tubule basically is like that, and then you have the fimbriae like that. Okay, and that's the ovum. Now inside this there are the cilia, and the cilia will all move like that inside. You know, when it moves inside, it is creating a negative pressure inside. It is this negative pressure that allows the fimbriae, which is actually moving over the ovum, which is sweeping over the ovum, and so immediately the ovum gets engulfed into the ampulla of the fallopian tubule, and then at the junction between the ampulla and the isthmus, that is where the fertilization occurs. Okay, so important point to remember that the fimbriae sweeps over the ovum, and the movement of the oocyte into the uterus is created by the cilia of the oviduct that create currents that sweep over the uterus. Fantastic. So this will complete this section of the questions. We are going to be doing 100 questions. बच्चे लोग almost 100 questions मारती each chapter ಅಲ್ಲಿ. So that you get a complete understanding of this one particular chapter. So go chapter by chapter, keep reading other things, but daily I want you to come back to this chapter and revise it. It's like that. So you're reading other chapters, but at the same time come back to this chapter, see these videos in short, just short videos. So while you're going in the bus, while you're traveling, while you're having your lunch, while you're basically resting in between, these are the times when you should, or you're bored of studying, in between 10 minutes, just see RN NEET questions and RN NEET shorts. You will capture a lot of information, and you will know where you know, what is the point you know, what is it that you don't know. Keep remaining logged in, study well, we will crack NEET like a topper. Okay, see you all. Bye.